Hello, in this video, I'm going to show you a concept in molecular biology known as central dogma. Central dogma means how information is moved from DNA to RNA, then RNA to protein. So I'm going to use BioPython to explain this. So let's import the necessary libraries from bio.seq import seq. So this one is called sequence. Seq means it's going to hold our DNA sequence. So we're going to create a DNA. So the DNA, we're going to use the seq to hold our DNA sequence. So I have already a sequence, so let me paste it. This is the DNA sequence we are going to use to create the RNA and the protein. So let's print our DNA sequence. Print, this one is going to be named DNA sequence. So sequence means the arrangement of the nucleotide. So let's print. So here is the DNA sequence. E stands for adenine, T thymine, G guanine, C cytosine. So let's create an RNA from the DNA. So let's create RNA. That's messenger RNA. So I'm going to call it mRNA. Don't mind, or it should be in small letter M, then the RNA in capital letters RNA. So let's create. So we're going to use the DNA to create the RNA. But okay, let's just go. I wanted to create a complementary. So we're going to write DNA dot. The BioPython has a function known as transcribe. So this transcribe will convert the DNA to RNA. So this one will be called messenger RNA. So let's print our RNA and see. So we're going to use the print in small letter print. This one is going to be mRNA, comma mRNA. So here is our messenger RNA. You can see the difference between DNA and RNA is that thymine is replaced by uracil. So let's go. Let's create our protein from the RNA. So we're going to use the messenger RNA to create the protein. So we're going to use mRNA dot, like again BioPython has another function known as translate. So it translates the RNA into protein. So we're going to name this protein. So this is going to be called protein. So let's print our protein and let's see. So this one is going to be named protein. Let's print the protein. Print protein. So okay, protein. So let's print the protein and let's see. Okay, look at the protein. Here is our protein. I know M stands for methionine. I don't know, maybe E arginine, glycine, histidine. I don't know. I used to carry out, if I see a protein that I'm not sure of, I used to copy all the protein and ask ChatGPT to tell me. So let's go there and see what ChatGPT has to offer. So I'll paste it and I'll run it and see what will happen. So like ChatGPT is telling me that M is methionine, A is alanine, G is glycine, H is histidine, R is arginine, valine, histidine, okay, Y is tyrosine, and L is leucine. So let's go back. So I want to print everything close to each other so that we'll see how DNA is moved to RNA and RNA to protein. I wanted to create the complementary base pair, but the complementary base pair is, so let's print them. You can see the DNA, the messenger RNA, and the protein. So let me just print them closer to each other and let's see. Let me print them so that they align with each other. So let's see. I will use formatted string. So let's try. I'm trying to make them appear close to each other so that it becomes easy to understand. So let's see, there's a problem here. Where is the problem? Is it from the comma? So let me put this one here. Let me put this line here. So why is the first one right? Is there something that I didn't do perfectly? All of them are inside the, okay, I didn't include the formatted string. So let's print it and see. You can see them. Let me move them closer to each other. Let me move this protein also closer to. Let's run them and see. You can see them. If you are smart, you can see how the DNA is converted to messenger RNA. So how RNA is converted to protein. So let me print complementary sequence. I don't think it's just normal. Basically, this is how to carry out central dogma from DNA to RNA and RNA to protein. Thank you.